a very warm greeting to one and all. My name is Shrikari, and I would like to welcome you all for the orientation session for OmicsLogic BioMMED Fall training programs. And this fall, we are excited to conduct programs on Introduction to Multi-Omics, Genomics, Metagenomics, Transcriptomics, and Biomedical data science using Python. So before we begin with today's orientation session, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And we have had the opportunity to work with coordinators from different parts of the world to provide bioinformatic training programs in different specializations, including oncology, infectious diseases, precision medicine, neuroscience, and biomedical data science. Now, the key features of the program include an industry-relevant modern asynchronous curriculum with both code and non-code modules. You'll be guided by an expert bioinformatics team and gain hands-on experience with essential bioinformatic tools, algorithms, and data repositories. And at the end of the program, you'll have the opportunity to work on a research proposal and receive expert feedback. And this has been achieved through a combination of online lectures, bioinformatic data analysis tools, curated data sets, practical assignments, hands-on coding exercises, and interactive mentor sessions. Now, to further enrich the training curriculum, we also provide real case studies that demonstrate the practical applications of bioinformatic methods and omics data in fields such as medicine, biotechnology, agriculture, and even astrobiology. Now, as I mentioned, during the training program, you will be given options for analyzing a given dataset. These include code-dependent pathways or code-independent pathways. Now, if you're someone interested in opting the non-code pathway, they will be given directions to access the T-BioInfo Platform for bioinformatic processing and analysis of data. And the platform includes demo pipelines as well as data management and analysis cloud infrastructure to run different bioinformatic pipelines on genomics, transcriptomics, metagenomics, and so on. And different stages of analysis are performed in different sections on this Multi-Omics platform. We will be covering about this in a short while. And at the end of the training program, you will acquire a comprehensive skill set, including a deep understanding of biological concepts, as well as the processes governing their interactions. You will also develop strong computational skills and proficiency in programming languages such as Python and R. You will also gain expertise in statistical methods to analyze large omics datasets. So now to view the various courses example projects, you need to first start by signing up on the OmicsLogic Learn portal, where you will be getting access to the training resources as well. So here's the link for signing up. And this is for those participants who have not signed up on the portal yet. If so, please follow along. I'll just walk you through the steps on how you can get started. So in my email, I've mentioned the steps for signing up, and here's how you can do the same. For those of you who have not signed up, click on the Create an Account option that you see here, and you'll be asked to enter few details such as your name, email ID, password, your university, and so on. Make sure that you log in using your university credentials. And if you already have an existing account on the portal, you can directly click in Login Now and enter your existing email ID and password and sign in. So with that, I'll just pause for a couple of seconds. If the participants have any queries about signing up or if you're facing any technical issues, we'll address them and then proceed forward. So I'm monitoring the chat box. Please put a yes in the chat box if you have already created an account on the portal so that I get an idea. Okay, thank you so much. I have received one response. What about the rest of you? Have you also signed up? Or if you want me to walk you through once again about how to sign up, please let me know and I'll be happy to do that. Thank you so much, Dr. Maria. We'll email out the steps once again so that if participants are facing any issues on how to sign up, you can just follow along the steps. So for now, I'm just proceeding with signing up and how you can get started. All right, so once you sign up, the very first thing that you have to do is to set up your account on the OmicsLogic Learn portal. To do that, head over to the blue bar on the top that says Welcome Back with your email ID. Click on that and it will directly take you to your profile settings. Now you need to have a completed profile to start with the program. And to do that, you can click on this profile button that you see here and you'll be asked to enter a few details. Make sure that your name is in proper capitalization and with proper spelling because this name will be printed on your certificates once you complete the training program. And add a brief bio about yourself that will consist of your educational background and some of your research interests as well. And you can also add a link to your social media handles. In my case, I've added my LinkedIn profile and ID. And also add a profile image. It could be your own image or it could be the logo of your university as well. So once all of these features are updated, it should be showing 100% profile completion. Now, during your training program, we will be monitoring your learning activity in this activity log as displayed here. And we'll be reaching out to you in case we notice that you're being inactive or stuck somewhere. And now under the courses tab, it will display the various coursework progress that you're making in the training program. And this is one of the important parts that you have to keep in mind. When you head over to your profile and you click on the program tab in the white bar that you see here, it will display the program that you are currently enrolled in. So once you click on this, it will directly take you to the program page where you will be able to get started with watching the lecture videos and accessing the various online courses. And now under the certificates tab, whenever you complete a particular coursework, you will receive a separate certificate for that, as well as once you complete each of the levels of the training program, you will be receiving the relevant certificates for that as well. So that is all that I wanted to walk you through about with getting started. And now to get started with each of these training programs, what are the various resources, what are the curriculum, I'd like to pass on the stage to my colleague Sonalika Ray to get started. Over to you, Sonalika.
Thank you so much, Shrikari. And hello, everyone. A very warm good morning to you all. Before we get started, before I take you through the program, what is the program, like what are the different types of programs that we are offering this fall, and also what each and every program consists of. It would be great if we could first of all hear from each one of you, what is your background and you're from which lab and what is your research work, or if you're a student, then you're from which core or what degree you are pursuing right now. So let's start with Camilo. You can switch on your camera if you can, and please unmute yourself. Please introduce yourself. We would like to know what is your background. If you're a researcher, you are associated with which lab? If you're a student, you are associated with which university? What is your degree? And what motivates you to join the program? Camilo, can we hear from you?
Hello, how are you doing?
Hi.
My name is Camilo. I'm a biochemistry junior at LSU, and over the summer I did an internship at the Cancer Research Center, and I saw some bioinformatics work, and I was really interested in learning about it. So I'm more so on the experimental and like bench top research, but I wanted to know more about the software and the technology side of things.
All right. So have you registered for a program already, or you have joined to learn about the programs and then take up one?
I signed up for the Genomics program.
Okay, got it. All right. Next is Nimrat. Nimrat said she can't speak. You are a PI from University of Vermont. All right. What is your field, Nimrat? That would be great if you can tell me. Interested more about bioinformatics side of things apart from your expertise cellular biology. All right, got it. And which program have you taken up? Have you already enrolled or you wish to enroll now? You can put in the chat. Okay, we'll discuss about the programs, what we'll cover in each one of them, and then you can take a call. Thank you. Next is Dua Alawat. Can we hear from you?
Yes. Yes, my name is Dua. I'm a PhD student in University of New Orleans. My major is computer science, but I'm working in bioinformatics research. My dissertation is about infer gene regularity network using bulk data sets, bulk RNA sequence, and single cell RNA sequence data. I have issue with dealing single cell RNA sequence data, and I need to understand more about this type of data, and I use it in my research.
All right. Single cell is one of those areas that I'm very fond of to work on the analysis. Good to have you with us. I'll take you through the bulk and the single cell RNA-seq program, and it would be the best for you to take it up.
Thank you.
We have Dr. Maria with us. Hi, Dr. Maria. I know you, but others don't know you. So can you please introduce yourself?
Sure. Hi, Sonalika. So I am Maria Sanchez from LSU New Orleans. I've been with those guys already I don't know, 1 year and a half, maybe. So I've been learning bioinformatics, and I am working in cancer research and immunology, doing several things like RNA-seq and attack-seq, chip-seq. So I've been learning a lot. Thank you.
Thank you, Dr. Maria. And we have Ayan Shina with us. Can you please introduce yourself?
Hello, everyone. How you doing? So my name is Anthony Ayoshino. I'm with LSU School of Veterinary Medicine. I'm in the comparative biomedical sciences, and I'm having my PhD in biomedical science. So my background is in biochemistry from the University of Lagos in Nigeria. And right now I am working with Dr. Charles Lee in neural systems. That's the field under neuroscience. So I'm a first year PhD, and I'm very soon, maybe in my second year, I will have to develop proposals. So I'm looking for how I can incorporate knowledge in bioinformatics into neuroscience research. So that's why I have applied interest to do introduction to big data bioinformatics.
All right. We have worked on several neurodegenerative disorders. We have worked on Alzheimer's, Parkinson's, and Rett syndrome as well. So we have these case studies. I'll just take you shortly on the portal, and I'll showcase you these case studies. It would be very interesting for you to have a look at these different case studies using different types of data, RNA-seq data, and also metagenomics data for Rett syndrome. So I'll do that. Thank you.
All right. Thank you very much.
You're welcome. We have Ramesh with us. He's not able to speak, no problem. So you are a postdoctoral researcher, and you have worked on bariatric surgery, myeloid cell biology, and cancer, chronic inflammation. That's interesting. I've never heard about bariatric surgery before. I'll definitely look into it, what it is. Thank you so much, Dr. Ramesh, for introducing yourself. And Dr. Ramesh, have you taken the programs before as well? Because we had a lot of Dr. Ramesh with us. I'm not sure if you joined before. Are you new to OmicsLogic, or have you taken up a program with us before? Share my screen in the meantime. All right, so okay, Dr. Ramesh is new. Welcome. Okay, so I will take you all through the different fall programs that we are offering. I'll just discuss very briefly what all these programs are about and what will we cover in the programs and what would be the resources for the programs so that you can take up a decision which specific program you would like to join. All right, so I'll just take you through all of it now. And so this fall, we are offering 5 specific programs. So on the OmicsLogic Learn portal, you'll see that there are several programs around omics data analysis, which covers RNA-seq analysis, single-cell genomics, metagenomics, and also specialization tracks like infectious diseases. But this fall, we are specifically offering 5 programs, which includes Multi-Omics Bioinformatics to introduce non-bioinformaticians to the field of bioinformatics, because it's everywhere in all the fields right now. And it's very important for even all the biologists or medical students to really know about the field bioinformatics. And then we are also offering metagenomic data analysis, genomic data analysis, transcriptomic data analysis, training programs to understand how these different types of omics data could be analyzed. And a very important tool that is used to analyze different types of data is Python. So the last program that is up is the Biomedical Data Science in Python program. So I'll now take you through each one of these programs one by one. And these are some of the specifications about each one of these programs. So the first program is Introduction to Bioinformatics. Now, Introduction to Bioinformatics program has been designed for newbies or the ones who are from a non-bioinformatics field or biotechnology field. And you might have heard a lot about what bioinformatics is all about, and you might be curious of how do we really use it. So what is bioinformatics? Bioinformatics is the science of both storing lots of complex biological data and also of analyzing it to find new insights, which we use in many different ways. And today's biomedical researchers, the research around biomedical data isn't simply about publishing one set of results and hoping that other researchers read it. It's about finding patterns in all the forms of data, linking everything that is out there to provide new insights that we can only spot if we see the bigger picture. Bioinformatics lets us bring together the data from lots of experiments in one place, so we can ask those big questions and find the answers. And such data from different sources is available, and rather than the availability of such rich sources of data, it changes the nature of biological research itself, as it's rapidly acquiring the character of a data science. Now, who uses bioinformatics and who is it for? The answer might have been statisticians, computer scientists, or engineers, but the truth is that more and more biologists and clinicians are now expected to understand and handle their own data to publish and succeed in basic and translational research. In fact, biology itself is rapidly acquiring the character of a data science. So as biology is rapidly acquiring the character of a data science that I just mentioned, there are billions of data points on genes, proteins, as well as other molecules that are compiled in large files and they're systematically studied. And analyzing this data leads to more knowledge and understanding about living organisms, which includes human health, biotechnology, crops, and livestock. So in the training program, we will give you an overview of bioinformatics for understanding multi-omics data analysis. Now, when I say multi-omics data analysis, we'll give you an introduction to different types of omics data, be it genomics data, transcriptomics data, single-cell data, or metagenomics data, or even epigenomics data. So we'll introduce you to these different types of data resources from where you can extract these different types of data, what you can analyze with the data, and how do you do that. But the program is not in-depth analysis of each one of these different omics data types, but we'll just give you an introduction. We did not want to make an introductory program much heavy, and that is why we have specialized courses that you can pick to understand how specific analysis related questions could be answered, be it from the field of genomics, transcriptomics, or metagenomics. So I just wanted to mention here that we have these different types of omics data that we have introduced in the program, and we will also discuss about the different types of tools and techniques that we could use to analyze this data, to analyze multi-omics data. Then there are several data repositories that we could leverage from. I understand there are several PIs who are generating data in their lab, and they have the access to the data for analysis and have their research work done. But there are students who want to learn how to extract data from public repositories, and also learn how to perform different types of analysis to answer a specific research question. So there are different types of repositories from where you can extract the data, you can pre-process the data or clean the data, and then analyze it to extract meaningful insights out of it. So in the program, we'll introduce you to different repositories or databases, I should say, from where you can extract different types of omics data. So the program is designed to highlight the role of big omics data, that is shaping the modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, as well as machine learning that is now transforming personalization of treatment selection and innovative algorithms that allow for unprecedented capabilities with drug discovery, design of large molecules, and even drug repurposing. So we'll cover some of the important technologies that I mentioned with its applications in today's world. And the participants will be able to access bioinformatics resources along with expert and mentor support to get help with the learning process that will help you learn the transformation of biology to data-driven biology. Then let's discuss about the next program, that is genomic data analysis. So whether you are just starting your journey or looking to expand your expertise, this program is designed to provide you with the skills and the knowledge that is needed to excel in this rapidly evolving field. So there are three different levels that we have designed for all these programs. The first is the beginner level, then we have the intermediate level, and then we have the advanced level. In the beginner level of genomic data analysis program, you will develop a strong foundation exploring bioinformatics, NGS, and gain an ability to explore and even analyze genomic data through data exploration and also through sequence alignment, and there are many more different types of analysis that we would be doing. So from advancements in NGS to understanding the entire workflow from sequencing reads, quality control, alignment, and annotation, which files are involved, what it includes, how to prepare them, and what are the steps of analysis, what parameters to use, and finally, how to annotate the variants that are observed when the reads are aligned on the reference genome is what we'll cover in the program, in the beginner level of the program, and we'll further discuss more in the intermediate level that will take you deeper into the intriguing topics such as variant calling. We have a dedicated pipeline on the T-BioInfo server for variant calling. I'll just take you through that as well. We'll also discuss about copy number variation analysis, machine learning, genome-wide association studies, and ethical considerations as well. And for those who are seeking advanced knowledge, we will delve into population genetics, DNA sequencing analysis, VCF file analysis, functional annotation, and also we'll discuss all of these different concepts with real-life case studies on SARS-CoV-2, breast cancer, oncogene variant calling, and even Ebola. So by joining this program, you will gain a practical hands-on experience. You will learn cutting-edge techniques, and you'll unravel the mysteries that are hidden within the genomic data. So be prepared to unlock a world of exciting possibilities and make significant contributions in the field. These are some of the research projects that we have worked upon. So once you complete the training program, you can have a research proposal ready and further work on an independent research project. If you're a student, it would be helpful to you to submit it in your college as a dissertation or have a publication for higher studies. So that was about the genomic data analysis program. And for students, they often look for the potential career pathways. After being trained with a specific type of analysis, the question is, what specific career pathway can we choose now? Where can we apply the knowledge that we have gained? So once the programs are towards completion, we'll also have a session, a discussion on different career pathways, and we keep on posting different jobs as well, job openings on the OmicsLogic Learn portal that I'll take you through. That would be helpful for you to pick your dream job. Now, let's discuss about the next program, that is metagenomic data analysis to unravel the microbial community. Now, to explore the limitless potential of metagenomic data analysis through our comprehensive training program, we have again designed these different levels, beginner, intermediate, and advanced. So whether you are starting your journey or seeking to expand your expertise, this program equips you with the skills and the knowledge to excel in the dynamic field. So let's quickly discuss what is metagenomics all about and where did it come from. So in the 1800s, the link between the disease and the microorganisms was established, shedding light on isolated bacteria functions. And today we understand that the human microbiome consists of structured communities forming biofilms with complex interactions, aiding ecological stability. And advances in metagenomics enables the study of the vast majority of uncultivable microorganisms within their natural environments. So this course would cover bioinformatics analysis of metagenomic sequencing data, which includes genomic sequencing technologies, data utilization, analysis tools, and also the real-world applications. Now, since 1990s, the microbiome studies have made significant progress, notably through initiatives like the Human Microbiome Project that has enabled metagenomic sequencing advances, allowing characterization of microbiota in various human organs, and it presents opportunities and challenges in identifying their roles in organ health and diseases. In this program, we will learn about the different types of metagenome sequencing techniques to different types of data that is generated, such as OTU sequences that is annotated by mapping on to a reference database, such as RDP, that is Ribosomal Database Project, Green Genes or the Silva database. And such databases contain taxonomic information for the domains of bacteria, archaea, and eukarya, based primarily on phylogenes for small subunit rRNAs, that is 16S for prokaryotes and 18S for eukaryotes. Now I'll skip some of the basics on metagenomic analysis. We'll discuss more about it in the program. And I wanted to mention here that in the metagenomics program, we'll cover two major pipelines. One is the data to pipeline, where we'll understand, this one is the taxonomy classification pipeline. And then we'll go to the data to pipeline that will help you denoise and correct the errors in amplicon sequence data and provide you with high resolution error-free sequence variants. We'll learn what the flow of the pipeline is, what the workflow or the steps involved are, what parameters to use for different types of sequencing that is done, and how to interpret the results. Data to pipeline will give you abundance tables, phylogenetic trees, and will also help you with the alpha diversity and the beta diversity as an output. And we'll also go through the kind to pipeline, which will help you analyze and process microbial community data, offering diverse outputs, again, like taxonomic assignments, diversity matrix, and various visualizations. Also in the metagenomics data analysis program, we will learn about using R for different types of analysis, So the pipelines that I showed you, behind them are popular coding languages such as R and Python. And we also have in the coursework tutorials explained in R to help you understand how you can use a computational script to analyze metagenomic data. It would include beginner tutorials on data importing, preparation for analysis, and also standard visualization of processed data. And there are more specialized packages with publication-ready outputs that will also be covered in more advanced lessons. So that was all about the metagenomic data analysis program. And if we talk about some of the applications or practical examples of metagenomic sequencing, so you will learn about the applications of metagenomic data analysis that would include studying about skin conditions, food sensitivity of the gut, autoimmune disorders, cancer, antibiotic resistance, drug resistance, and even ecological studies. We have several case studies that we have put together on the OmicsLogic Learn portal that I'll take you through to understand how the different concepts of omics analysis could be applied to several research questions in the real world. Last but not the least, I want to discuss about the Python program. Why Python program? Why biomedical data science using Python program? So we have put together this program as the demand for skilled data analysts and scientists who can derive meaningful insights from biomedical data has increased. It's never been greater. And that's where our online mentor guided training program on biomedical data science using Python comes into play. In today's rapidly evolving world of biomedical research, the generation of big data has become the norm, and every day there are vast amounts of valuable biomedical data that is being produced, holding the potential to revolutionize our understanding of diseases and also pave the way for groundbreaking medical discoveries. However, the sheer volume and the complexity of this data presents a significant challenge, and that's where data analysis comes in. So this comprehensive program is designed to equip you with the knowledge and the skills that you need to excel in the field of biomedical data science through a combination of practical assignments, live workshops, and online resources that you'll embark on a transformative learning journey that will empower you to unlock the potential of high throughput biomedical data. Now, in this program of biomedical data science using Python, we'll also introduce you to different types of omics data. Let's say you are a biologist, not from the field of bioinformatics, and you have little idea about these different types of fields, but not dealt with the different types of omics data. But when you have to learn Python to deal with biomedical data, it's very important for you to understand what these different omics data would really look like, how you have to deal with them. So we'll introduce you in the beginner level with all these different types of omics data. First of all, we'll also discuss how to extract this data, how to clean this data, and how to analyze this data, all with Python. So we'll start by understanding the basics such as data types, variables, control structures, functions, and modules as well. And then we will dive into data processing and cleaning, where we will use powerful libraries like NumPy and Pandas. And then we will learn about exploratory data analysis, leveraging visualization libraries like Matplotlib and Seaborn to uncover patterns and the relationships that are hidden within your data. Additionally, you will gain an understanding of dimensionality reduction techniques and machine learning using scikit-learn library. So as you progress to the intermediate level, you will deepen your understanding of clustering and advanced machine learning techniques that are specifically tailored for biomedical data analysis. You will explore clustering techniques like k-means, edge clusterings, spectral clustering using Python clustering packages like scikit and scipy. And then you'll learn model validation techniques and evaluation matrix for supervised machine learning models. You'll also delve into advanced machine learning techniques like decision tree, random forest, SVM, LDA, and UMAP, and apply them to real-world biomedical data sets. What are these different algorithms? How do they work? How do they create the machine learning models? And which one should be used for what specific type of research question? That is what we would be discussing in the program as well. And last but not the least, I wanted to discuss this because in today's data-driven world, AI that has powered a lot of technologies like ChatGPT, it is revolutionizing the way we approach scientific research and data analysis. And as learners, you have a remarkable opportunity to enhance or to harness the power of AI to unlock new insights. AI, particularly the code interpreter in ChatGPT, and now there are different AI tools as well, can provide assistance by generating code for complex analysis and automating routine tasks. Additionally, it can produce insightful visualizations such as heat maps or dendrograms and other plots that could help you visualize and interpret your data effectively. We know that we can generate code, we can understand a code, we can produce a code, and we can also create different types of visualizations now with the chat GPT's paid version that they have, that they mention as GPT-4. But when it comes to creating these visualizations, it's very important for you to understand the logic of the analysis. What logic to apply? For a specific type of visualization, what prompt you have to use that you could input in these AI tools to get the desired result that you can use in your research publications. So in the training programs, we'll be going through all of these different techniques. We'll discuss about the logic for different omics analysis that you have to use through which you can generate these interesting visualizations and plots. So that was all that I wanted to discuss about the different training programs. And now I'll take you through the OmicsLogic Learn portal, where I'll showcase you different resources that you have an access to, as a part of the program. So, Srivari, can you just confirm if the OmicsLogic screen is visible and not my slides?
Yes, we can see the program.
Thank you. All right. So as Srivari explained you about the OmicsLogic Learn portal, I hope you all have signed up now. If you have not created an account yet, please feel free to create one. I'll put the link in the chat. Now, on the OmicsLogic Learn portal, once you have signed up, there are two tabs that are very important for you. The first one is the Programs tab, and the another one is the Courses tab. Now, on the under the Programs tab, you'll see a lot of omics analysis and research specialization specific training programs that you can enroll and learn from. I'm going to discuss about these 5 programs that I took you through. The first one is Introduction to Big Data Bioinformatics. Another one is Transcriptomics for Biomedical Research. Then we have Genomic Data Analysis, Biomedical Data Science using Python. We are also offering Chem Informatics. If anyone is interested, you can let us know. And then you have on the second page, you have Metagenomics. You can log into the portal, go to the Programs tab, and feel free to go over the curriculum. And once you have joined a specific program, so once you register for a program, we enroll you on the program page, and you'll find a badge of the same program on your profile. So you can also access the program that you have enrolled for through the program link, but you can also access it through the badge here. So if you have enrolled in Transcriptomics, you'll have a badge here. If you click on it, you will get redirected to the program page. Now, let us understand what the program page is for and what resources can you leverage. So once you are on the program page, you can see 3 different levels that I mentioned. The first one is beginner, then you have the intermediate level, and then you have the advanced level. The beginner level has 6 sessions, intermediate level also has 6 sessions, and advanced one has 12 sessions. As a part of the program, you have the access to the intermediate level, but if you wish to extend it to the advanced level, you can reach out to us and we'll help you with the extension. Now let us see what are the components of each session. So if you click on the drop down button, you'll get the sessions. Now for each session, you have different sections under which you have a session video. It's an embedded YouTube unlisted video that you have to go through. And then you have an associated lesson. I'll just go through this, what it is. And then you have the additional reading material. It could be a reference paper, a reference book, or even an interesting video that could help you understand the concept through animations. So that is what you will get in the additional reading material. And in some of the sessions, you will also get the link to the demo pipeline. So if in a session, the mentor is explaining how to perform or how to process or execute a pipeline on the T-BioInfo server, you'll get the link to the demo pipeline as well. And the link to the practical lecture would already be there under the section of the topics, where you will learn how to process the pipeline, the basis, the workflow, the parameters to use, and what results you get, how to interpret them. And for if you're interested to run the pipeline or execute the pipeline yourself and check how to do so, you'll get the link of the data as well that you have under each session that you can refer to. You can see what the data structure looks like and also run the pipelines. But we recommend that you go through the demo pipelines where the data is already uploaded. I'll just show you one, and you can execute that pipeline because it's quick. And the motive here is to understand the flow of the pipeline and also to look at the results and understand how to interpret them. So demo pipelines are easy to learn, quick to learn. And once you have your own data, you can then process the pipeline with your own data and interpret your results. Now let's go back to the component of associated lessons. Some of you who are joining us for the second or third time would know how to access the program page, but I'll go through this for the newbies here who have joined us for the first time. Now, what is an associated lesson? An associated lesson, you can click on it. It will redirect you to a lesson of the coursework. So for all these sessions that we have put together here, you have a coursework or a lesson that you have to go through. Now, from where these courseworks come from? This is the courses tab. I'll just take you to that lesson link that I opened, but meanwhile, I'll tell you from where did we get the courses. This is the courses tab. Now, if you are a part of the Introduction to Bioinformatics training program, you will have the access to some introductory courses. Now, what access of which courses do you have? On the training program page itself, I think my internet is slow, apologies for the same. On the training program page itself, if you scroll down, you'll see a program, course, and project library. So as a part of the program, you'll have the access to all of these courses that you can go through. You can click on them, they'll redirect you to the course. You can also search for the course here. So if this is the course, a course has several lessons inside it, and you need to complete each one of these lessons. So if it's Introduction to Bioinformatics, in this course, we're introducing you to different types of omics analysis, be it genomics, transcriptomics, or metagenomics, and also the basis of bioinformatics. Inside each lesson, you'll find the concepts through theory. You also have videos to watch, and you also have quizzes and assignments to answer. So that is what constitutes a lesson. Now, this lesson would be linked to each one of the sessions on the program page. So if you're going through the first session, you have to complete the first lesson of Introduction to Bioinformatics coursework as a part of the session. Now, not to complicate things for you, that's why we have put everything on one program page. You open the session, you watch the lectures, you go through the associated lesson, you read the additional material that we have shared, and proceed ahead. Now, once you complete the session, you need to click on the complete session button as well, so that it's marked on your profile that you have completed this session. And when you complete the 12 sessions of the program, only then the certificate would be issued to you on your profile. You'll get it automatically on your profile from where you can download. So that was about the lectures that you need to go through and the associated lessons and the reading material. Now, for introduction to bioinformatics, I mentioned you have the beginner, intermediate, advanced level. And this was the lesson link that I clicked on just to show you how it redirects. Now for transcriptomics, I did not discuss about the transcriptomics program in my slides. So I'll give you a brief overview about this program through the program page itself. In the beginner level, we'll discuss about different NGS. We'll discuss about different techniques of NGS. We'll discuss about the different types of sources for data extraction, different repositories, and this is specific to RNA-seq data only in the beginner level. And then we'll discuss once you have the data, let's say the data is FASTA files or FASTQ files, how do you process it to generate a gene expression table? What other tools involved? How to perform exploratory data analysis? How to perform downstream data analysis, which would include differential gene expression analysis, pathway analysis, functional enrichment analysis. And in the intermediate level, we'll discuss about pathway annotation. We'll discuss about visualization and validation of the results. And we'll also discuss about regression and factor regression analysis and introduce you to machine learning. For machine learning, we'll discuss about unsupervised and supervised analysis. Now, both of these levels, beginner and intermediate levels, these both levels have the practical hands-on with regards to the T-BioInfo platform and also open resources. When I say open resources, I'm referring to the databases and the tools or the softwares that are present openly that you can download or maybe explore if it's a server. So we'll discuss how different tools online could be used for RNA-seq analysis as well, along with the pipelines on the server. And in the advanced level, we will work with computational languages R and Python to analyze RNA-seq data. So the process would start from the very basics on the beginner level, how to import data, how to perform exploratory analysis, statistical analysis, correlation and regression, differential gene expression analysis, pathway analysis, and unsupervised, supervised machine learning. And we had a participant who was interested to learn about bulk and single cell analysis. That's why I wanted to explain the advanced level here, where you have sessions on single cell RNA-seq analysis. We here discuss about the single cell analysis workflow, what tools to use, what parameters to use, what the results would be, and how do you interpret them. Once you have the results from the pipeline, you have to perform further downstream analysis for cell identification through cell clustering, and that is done through R. So how do you do that? And you'll learn everything step by step in the RNA-seq analysis. And those who extend their program to the advanced level, towards the end of the advanced level, you'll get to work on a capstone project as well, where we'll discuss your research proposal and take it further as a research project. So that was about the transcriptomic data analysis program. And as a part of the program, you have the access to several courseworks that you can go through. Transcriptomics using the server, transcriptomics in R, and transcriptomics in Python. I'll explain you the specificities of R and Python coursework through the Python program so that you know what is included. Because of the time limitation, it's not possible for me to go through each one of the courses and showcase you the topics that we are covering. Please feel free to create an account and you can explore the OmicsLogic Learn portal and see what all topics are there. Now for the genomic data analysis program, we have the 3 different levels specified here. Please take a while to go through the curriculum while picking up the coursework that you would like to pick from. And then comes the biomedical data science using Python program. Now, for the Python program, there's definitely code block that we need to practice, right? So for that, we have put together several Google Collabs. And on the program page itself, you will find the link to the Google Collab. So this is the Google Collab that you have to refer to for what is also explained in the session videos. And you have to create a copy of this Google Collab and then practice with the collab, practice your code blocks. And if you get any errors, you need to reach out to us. And we prefer that you work with the Google Collabs because it's easy for us to check your errors, help you with your errors, and easy to process with. So that was about the Python program. Now let's quickly go through the T-BioInfo platform for those who have joined us for the first time. So this is the T-BioInfo platform. Meanwhile, at logins, I also show you the Metagenomics program. It got skipped. So the Metagenomics program similarly has these 3 levels, and you can go through the curriculum here, and then you have the coursework and the case studies. Case studies is one of the things that I would like to discuss. Let's go over the pipelines first, and then let's see what case studies are that you can learn from. So this is the T-BioInfo platform, where you have these different areas of analysis: transcriptomic data analysis, genomic data analysis, cancer bio, single cell, metagenomics, virology. And here's a section for machine learning, GWAS, and phylogenetic analysis, and differential expression as well. Now, when I mentioned demo pipelines, the links of the demo pipelines that were present on the program page, those are basically these demo pipelines. These are basically the pipelines for specific case studies on the portal. Now, when I say case studies, under the courses tab itself, if I type transcriptomics, along with the coursework, I'll get several case studies as well. These are case studies. Now, these case studies are for you to understand how to apply the concept that you learn in the coursework to real life research questions. And the coursework is quite lengthy because we are discussing about each and every step from the very basics to the advanced analysis that would be required. And just one thing, a lot of participants get confused with the lessons. There's a button called load more here that will load all the lessons. So don't miss out on the lessons. Now, the case studies are not very lengthy. They are short case studies with an application of the concept. So you should go through one of the case studies at least to understand the application. Okay, now for each case study, you have a case study pipeline here that you'll get under the drop-down button. I'll open one of these case studies pipeline to showcase you what it includes. Now it's named demo just because the data is already uploaded at the backend. It's a pre-processed pipeline. You need to process it just to look at the algorithms that are being used and understand how to interpret the results. So this is a differential gene expression and analysis pipeline, which takes the data and you upload the file, the raw rate count file with 2 groups, and then you use these different algorithms for differential expression analysis and pathway enrichment analysis, functional enrichment analysis, and you get your results. The motive behind processing a pipeline is definitely one is it's quick, it's efficient, it will give you your results in a shorter span of time, and it gives you all the results, all the possible results. It's very important for you as a learner to understand what these results are and how to interpret them. So we have 2 different types of results. One is the interactive visualizations that you can open and change the parameters, play with the parameters, and then learn from them. And then you have these files that you can directly download. These are zipped folders. You can download them, unzip them, and then look at the results. Because it's an orientation session, my motive is not to explain each and every result, but just to showcase you how easy it could be to process these pipelines, get your results, and also interpret them effectively. And you have these interactive results that you can explore and extract meaningful insights. Because this was a demo pipeline, I got the results the moment I clicked on the end button. But if you run the pipelines for your own analysis, it would just take few minutes to few hours to process the data and give you your results based on, again, the sample size and the file size. So this is a heat map where you can look at the differential gene expression of different samples. These are some of the top differentially expressed genes. And then you have a volcano plot. Again, you can interact with the volcano plots, look at the position of the genes, what is the fold change. And then you have a dashboard. In the dashboard, you can change the parameters and see how your results change. So if we are working on a research project, so experimenting with the parameters would be one of the useful things that you could do from a dashboard. You have a heat map here as well. There's something wrong with the name of the sample. It should be short though. But yeah, it's a clear heat map here. In this case, it's short, but you can change the log fold change and see how the heat map changes. Top genes line graph. And you also have the pathway analysis result here, which pathways are affected, how many genes are there, and then you also have the code block for, you have the data here, you have the symbol IDs, the annotations are there, the gene symbols, and the annotation is here, the calculated statistical values are there, and then you also have the code block that you can download and maybe practice yourself once. I downloaded the results here. I'll just show this to you. So one, we have the plots that you can download. This is a volcano plot with the mentioned gene names. For publications, obviously you have to refer to this volcano plot. The interactive one is for you to extract insights while changing the parameters. You can unzip the file here and then look at the results that you get. You'll get detailed Excel sheets and that you can export. This is just a differentially expressed file where you have the calculated statistical values. And then there's a lot more to explore. So as a part of the program, you'll get the credentials to the T-BioInfo platform. Take some time out. You can click on the drop-down button. You have several demo pipelines here that you can learn from. See what type of results you get and how to interpret them. The interpretation is mentioned in the coursework, so it would be helpful to you. So that was all that I wanted to discuss today. And if there are any questions, please feel free to unmute yourself and ask. I hope I did not bore you all, but it was quite an interesting session. I see Robert has joined us. Hi, Robert. I did not hear from you. Could you please introduce yourself? What is your background? Are you a researcher, a student? You are in which lab?
Hi there. Yeah, my name's Robert. I'm a graduate student at UNLV looking into, and I wanted to take this course because we're getting into RNA-seq and kind of like more transcriptomic stuff. So I wanted to take this course to kind of brush myself up on those skills.
All right. Okay. So I hope you did go through the program page of Transcriptomics when I was explaining. Were you there or you joined after that?
No, yeah, I saw that.
You saw that. All right. Perfect. So feel free to go on the program page. And one more thing, I think we did not add the Transcriptomics program in the flyer of the NAPI offered programs. So just wanted to let you know that this program is also up there because we got a lot of requests for this program. So we had it up for the fall as well. So take a while, go through the curriculum, and then we can get started next week. Are there any questions?
Yes, Anamika. Dr. Chatterjee has put his queries in the chat.
Okay. In order to access the beginner or advanced modules for transcriptomics or basic bioinformatics, will we be paying? Okay, Dr. Chatterjee, are you here? Yes. Okay. So I don't think that's the amount that you need to pay. There's a specific scholarship on that. Ojasvi, can you please take up this question? I'm so sorry. I'm very bad with numbers. I wasn't trying to explain the conceptual things. Ojasvi, can you take this question, please?
Yeah, hi.
Yes.
Can you just repeat this question? I was just typing
Okay.
something. So,
Dr. Chatterjee is asking, do they need to pay the total $600 for each of those if she wants to get
programs.
the access to the beginner or the advanced modules of the program?
Like two different programs or one program? Because I see Dr. Chatterjee have already enrolled in Genomics, but if she wants to change to, let's say, transcriptomics or Python, or if she's interested in both, so we can give her like 10% scholarship and enroll in advance. I will like send the pending invoice because some amount is already paid. So for NEPI participants, there is a discount.
Okay, so
Yeah, same
Dr.
for
Chatterjee
Robert.
Ojasvi is the best person to contact for the program's enrollment.
Yep.
Ojasvi was saying something, sorry.
Yeah, and I guess Robert has also enrolled in Introduction to Bioinformatics, but if he is interested in transcriptomics, so he can change his enrollment, or maybe he can enroll in both, and I'll see the invoicing accordingly. Like I have their emails, and I guess we are already in touch through nepi dot this email ID. I'm just sharing in the chat.
Yes.
Station.
For any enrollment related issues, you can reach out to Ojasvi. He has shared his email ID in the chat. And this is for the ones who are from NEPI and the ones from LBRN, Ojasvi. Where do they
Yeah,
email
sure.
the same email ID?
Yeah, they can email me on nepi one. Yeah.
Okay. So
Or
everyone,
this is my
anyone
another
can.
email. So anyone can reach out anywhere.
All right. Thank you. And for resources, let's say, so every week you will get the resources on your email as well. All right. The resources would be updated timely on the program page also, but you'll also get it on the email. In case you don't get it, please check your spam folder. Make sure we are in your inbox and not spam. And if there is any resource that you need or you missed on something, or if you have any question, please reach out to us on communication@OmicsLogic or sorry, Pine.bio. Communication@Pine.bio. You, Sri Gauri, looks after this email. And if you have any conceptual question, you can reach out to me directly at sonalika@pine.bio. And we are planning to have live Q&A sessions. There was a time issue last time. So we were meeting some of the participants at 9 a.m. CST and some of them at 1:30 or even in the evening at 7 p.m. CST. So because you have enrolled in the program, we need to stay in touch for your questions, for your doubts. Please let us know what time works for you. Yes, we can do a doodle poll. Ojasvi suggesting we can do a doodle poll to understand what time works for you. And if we have two different cohorts for morning or evening, we can have two different meets. That's not a problem for us. Yes, we will share the recording with you, Dr. Nimrit. Yes. All right. Thank you everyone for joining. If there are no questions, I think we are good to end the call. And Dr. Maria, your query for Python. Actually, we were having up the practical sessions for each algorithm. each supervised ML algorithm. That's why the session got delayed. But by the end of today, you should receive all these different sessions. It is little heavy where we are discussing about each algorithm. So we wanted this session to be very much detailed, like in depth. That's why it got delayed. So we'll share it with you today and you can go through it.
Okay, thank you.
Welcome.
Thank you
And
so much.
about your analysis, we'll also get back to you. We are working on it. Yes. Thank you everyone for joining, and we'll see you next week. We'll have the sessions shared by next week, and let's get started. Thank you. Bye-bye.
Thank you, everyone.
Thank you, Sri Gauri. Bye, Yojasvi.